Hi there. Welcome to this pre-recorded lab session that will serve to supplement our weekly lab. I hope you are enjoying this spooky season and are getting to make most of the fact that we will not get to meet in person today on Wednesday, October 29th. Instead, I ask you to watch this video and also utilize other materials that I have shared at the Blackboard. This way, you will still be able to complete our weekly lab activity. So let's have a look of what you need to do to successfully complete this lab. Well, the first part, watching the instructional video, you are already doing it by being here and viewing this recording. Well done. The following steps you will complete on Blackboard. There, you can download the worksheet for this lab. Within the content area of our Blackboard course, go to the week 10 plus 11 module. Once there, scroll all the way to where it says Lab Inheritance. Click on it. This gets you to the lab that we are now working on. Here, you will need to click on View Instructions button, which is located all the way to the right on your screen. This initiates the lab. Now, you will see also the lab worksheet. You can download it by clicking the three circles next to the file and choosing Download. This gives you a Word document that you can edit. You will already have an access to Word as part of your student account in our institution. If you prefer to print this document and then write on it before uploading your completed scanned file, you can certainly do so. There are both printers and scanning access available on the campus. So the worksheet will provide you with all the information that you need to be successful in this lab. Make sure to take time to read through it. Then, once you get to page 6, the worksheet will ask you to collect group data on certain traits as table 1. I realize that we will not be able to do that since we are not meeting in person, but for some traits you will be able to provide your own data. I am not asking you to figure out whether you are PTC taster or not. You can skip that part. But you can tell me what kind of a hairline you have. Is it straight or does it have a widow's peak? Tick under whichever applies to you. Leave the other box empty. Same goes for dimples. You can indicate whether you have those or not. And again, same for whether you have free or attached earlobes. I am not asking you to respond on photic sneezing or whether you have astigmatism. But again, you can tell whether your hair texture is. Is it curly or straight? Or perhaps it is something in between the two, which would be known as wavy. Finally, no data is needed from you regarding color blindness or blood type. If you know any of these additional information, you certainly can include it, and I will be sure to add you extra credit for the additional effort. For Table 2, you are asked to provide the class data on these traits. How to do that? Well, lucky for us, I have asked my other class to help us. You will be able to find the class data again under this week's learning module. Look for class data on inheritance from Wednesday morning group. It is the very last item on this learning module. You will find there both numeric and percentage data. It is the percentages that you will record to your table 2 on the worksheet. Then, on page 8, you will have some questions regarding analyzing the results. You will be able to answer these as long as you have collected the class data. But as usual, I am here to help you with the trickiest ones. Let's have a look at those. On question 3, the worksheet asks you about incomplete dominance. This is an interesting pattern where the dominant allele does not completely block the recessive allele, which would be normally the case. Instead, heterozygous individuals express a phenotype that is somewhere in between the dominant and recessive phenotype. A good example of this would be hair texture. Then, the pattern known as codominance is also interesting. Here, there are multiple alleles that are dominant. As a result, we can see some heterozygous combinations where an individual expresses both of the dominant phenotypes together at the same time. A good example of this is ABO blood types. Both A and B alleles are dominant, and O is a result of 2 recessive alleles. But here, blood type AB is a mix of both dominant A and dominant B allele. Finally, while we have been so far looking at traits that are carried on chromosomes 1 to 22, known as autosomes, there are some traits that are carried in sex chromosome, which was our chromosome 23. Examples of these include red-green color blindness, where there is reduced ability to distinguish between the shades of red and green. Hemophilia A, which is a blood clotting disorder. DMD, a progressive muscle weakening disease, and many more. These are interesting for us because males have only one X chromosome, so they will express an X-linked trait if they inherit a quote-unquote bad chromosome. So carrier mother has a 50% chance of passing it to each son that they produce. So this brings us to the end of part 1 of this lab. But there is a little more to go. In part 2, which starts from page 9 of your worksheet, you will be looking at corn cobs. We actually know quite a bit about their genetics. A few pieces of information that is going to be useful for you here. We will look at 2 factors on the corn cob: the color and the texture of the seeds. It is going to be helpful for you to know that purple seeds are actually dominant trait for the corn, while yellow ones are recessive. We will mark allele for purple as capital C and allele for recessive as lowercase c. We will also look at the texture of the corn seeds. Smooth kernels are dominant trait and symbolized in our shorthand notes as capital T, while recessive trait is wrinkled seeds, and it is symbolized with lowercase t. But let's just focus on the first of the color. Here you will see a cross of what is known as the true breeding varieties of corn. True breeding means that it is homozygous, so it has 2 identical alleles. So let's work our way through this. The parent, which is symbolized as A1 here, appears purple. Remember, the appearance of a trait was its phenotype. So for this one, it is purple. The genotype instead is referred to what alleles it has. Because this is a true breeding parent, we know that it has to be capital C and capital C. That gave us purple color. Now let's look at the other parent. This parent labeled as A2 here has phenotype of yellow. As a result, since we know that it is a true breeding one, its genotype has to be lowercase c. You will be asked to create Punnett square for this cross, so let's do just that. As you learned in the lecture, Punnett squares are a tool that we often use to predict the outcomes of a cross. So we put one of the parent's alleles to the top and one to the side. This allows us to start filling in the details to the Punnett square. So the alleles for parent A1 are capital C and capital C. We put those each in one box. And the alleles for the parent A2 are lowercase c and lowercase c. Again, we put those each in their own box. So here we have a start of our Punnett square. Now we need to solve what kind of offspring, both in terms of genotype and phenotype, these parents A1 and A2 will produce. Let's do just that. The process is that we move the allele to each box, either below it or to the right of it. This gives us an outcome. Let's see that in action. Okay, my drawing of the movements of these letters was probably not as tidy as it could have been, so here we see the outcome written in a more pretty manner. This is what I expect you to produce for question 5 on page 10. Now let's make sense of the phenotype and genotype of this cross. So the genotype for all of the offspring is capital C and lowercase c. This means that 100% of the offspring will have this genotype. And 100% of the offspring will have purple phenotype. That's what I'm looking for to question 6 on page 10. Now, let's move on to page 11, where we want to predict the genotype and phenotype for the next generation, which will be the F2 generation. So the parents for this generation will be the F1 generation, which we just worked on. Note here, these F1 parents are no longer a true breeding crops. So here, both of the parents are going to be heterozygous for their genotype. So they have one capital C and one lowercase c. And we mark it as such on the top of the Punnett square and on the side of the Punnett square. So that's what we have here. And now all we have to do is to write out the results of this cross. Remember, as before, the alleles move either down or to the side, depending on where they are located. I just did that earlier on and did not animate here, but you saw it there, how it happens. This is what I would expect to see from you as you produce the answer to question 7 on page 11. From this, we can make sense of the genotype and phenotype. And again, these should help you with the question 8 on page 11. Now, you are going to copy these phenotype numbers from the cross as your predicted percentage numbers to the table 3 on page 12. But we want to see how the predicted numbers hold to the reality. To figure this out, you will actually count 100 kernels from an actual cross that was done to these plants. To aid you in doing that, you have table 3. Mark whether the kernel is purple or yellow to the tally row. Until you have covered 100 kernels, then add the final numbers on the last row. If you did this correctly, they should add up to 100, right? Finally, you are asked to reflect on how the actual numbers matched with your prediction. If it helps, you have this picture as a larger file on your blackboard. Just go to this week's learning module and look for the file that is named as actual corn cobs for OL Lab to analyze, as the second last item on your week 11 learning module, as you can see on the computer screen here. You are going to do the same thing for cross B, which you will find on page 13 of your lab worksheet. I have also shown it here. These are true breeding parents, so both have 2 identical alleles. Let's look at that. So the parent B1 is obviously one with wrinkled seeds. So as you learned earlier on, the genotype for that is lowercase t and lowercase t. The parent B2 is one with smooth seeds. So the genotype is uppercase T and uppercase T. This should help you with question 11 on page 13. Now, what kind of children are they going to produce in offspring F1 level? Well, to solve this, we will do a Punnett square, and I have already filled in the parents' alleles for you here. The process follows as you learned earlier. You will fill in the data to the boxes, bringing down or moving to the right the letters as needed. And as you get something like this, I believe. Not surprisingly, the 2 breeding parents of opposite traits produce here 100% of the offspring that are heterozygous. So all of the offspring will have genotype capital T and lowercase t, and their phenotype will be smooth because that was the dominant trait. Again, this should help you with questions 15 and 16 on page 13. As I'm sure you guessed, we are now going to look at next generation of the offspring that are heterozygous. So all of the offspring have the genotype capital T and lowercase t, and their phenotype will be smooth because that was the dominant trait. And again, this should help you with questions 15 and 16 on page 13. As I am sure you have guessed, we are now going to look at the next generation, known as generation F2. As we concluded in the previous step, all of the F1 generation had genotype capital T and lowercase t, so that was heterozygous, and the phenotype was smooth. That was because the smooth allele was dominant. Therefore, we can fill in the Punnett square accordingly. And now, all we have to do is to fill in the Punnett square. This works just as before. From this, we can figure out that the resulting genotype and phenotypes, which are 25% genotype capital T and capital T, 50% capital T and lowercase t, and again 25% genotype lowercase t and lowercase t. For phenotypes, it is 25% wrinkled and 75% smooth. Remember, smooth was the dominant trait. You guessed it, these are the numbers, and these are the numbers that we would like you to copy to table 4 on page 14 as the top row. This is our predicted outcome for the cross B. Now you are going to see if this matches with real-world results. So again, I have provided you a picture of an actual corn cob from this cross, and you can find it on this week's learning module if you have a closer look at it. But again, you should count 100 kernels and record these results to your table 4. Again, on question 20, on page 15, you will consider if your predicted calculations match with the real-world findings. For a good grade, you may wish to consider here what would happen if you counted more than 100 kernels. Your results would likely become more accurate and match better with the predicted outcomes, right? Good job so far. We are ready to move on to our very last cross. Here we are looking at 2 traits at the same time. So we are paying attention to both color and texture of the resulting kernels. Again, we are told that parents C1 and C2 are true breeding. This means that for each of them, their alleles are homozygous, so the same. But first, let's look at the phenotype for this. And remember, we now want to see 2 traits. For parent C1, it is going to be purple and smooth. And for C2 parent, it is yellow and wrinkled. As for the genotype, this gives us, for parent C1, it is purple, so the genotype color is capital C and capital C, since those were true breeding and purple was the dominant trait. And we will also include the texture, which is capital T and capital T, since smooth was dominant trait and these were true breeding. For parent C2, it is going to be yellow, so lowercase c and lowercase c, and wrinkly, so lowercase t and lowercase t. The reasoning is the exact same. We are working with true breeding plants. This should help you with questions 21 to 24 on page 15. Now, we want to predict the F1 offspring. To do this, we will use Punnett square. This time, however, since we are looking at 2 traits, the Punnett square gets much more busy. For each of the boxes for the parent's alleles, we will fill in both the allele for the color and texture. So the size of the table doubles within true traits. Filling it will be, however, rather easy, as we have seen here. Not surprisingly, based on all that we have learned, all of the children in F1 generation will be capital T and lowercase t with the capital C and lowercase c. This in turn allows us to figure out the genotypes and phenotypes. These are for all offspring in F1 generation, genotype capital T and lowercase t, capital C and lowercase c. As a phenotype, this is purple and smooth kernels. This should help you to fill in the answers for the questions 25 to 26 on page 16. Now, this is where it gets more complex when we want to figure out results for the F2 generations. So again, we can fill in the parent data based on what we have learned in the previous cross, just like that. And now it's going to be the tedious job of filling in the potential outcomes of this cross. Let's do it. Now, that is quite a project. To make our life easier, I'm here going to show you only the phenotype information. Further, when I try to analyze results like this, I often find it helpful to use some color coding to make sense of the results. So what I have done here is that the phenotype is, if it's going to be purple, I have colored the box as such. And if it's going to be yellow, I have colored the box as such. Now, if there was a smooth outcome for the texture, the box will be solid color. But if it was going to be wrinkly, it has a little bit of dots as a pattern. From these, we can do fractions of the outcomes. So 9 boxes out of 16 are purple and smooth, and so on and so on. We can convert these proportions into percentages as seen here. And that, I hope, helps you with question 27 of the lab worksheet. One last time, we are going to copy our phenotype data into the table. This time it is table 5, and this will give us our predicted values based on the mathematical probabilities. And one last time, you will calculate the actual results from an actual corn cob where this cross was made. And again, you will reflect on how well the results matched with what we calculated. Remember that for full marks, a simple answer yes or no is probably not going to be enough. Give a rationale for your response. And just like that, you have completed the second last step of this lab. Only thing left is for you to submit your final work through Blackboard, just like you typically submit the labs. Finally, if you have made it this far, a massive well done. I wanted to finish off by giving a few tips on how to get high marks on this lab. Remember, high scores require usually more than just an answer that is yes or no. We are particularly interested in your reasoning, so give a rationale of why you would answer yes or no. I also realized that it may be difficult to analyze pictures of corn cobs when compared to doing it in person. If you're unclear about the color or texture, the texture is more likely to be the challenging one here, of a specific kernel, it is okay to skip it and move on to the next one. You will just need in total 100 kernels per activity. I hope that you have found this video helpful as you work your way through to this week's lab. I will see you in the class next week.